Welcome to the TOEFL listening section. In this part, you will hear one or more conversations in an academic or campus context, such as a discussion between a student and a professor or between two students. You may take notes while listening. Listen to part of a conversation. You will hear it only once, so listen carefully. After the conversation, you will answer questions about what you heard. Some questions will ask you about main ideas and details. Others will ask about implied meaning, speaker attitude, or how ideas relate to one another. Use both what is stated and what is implied to answer.
Good afternoon, Dr. Thompson. Thanks for meeting with me during office hours. I wanted to go over the vaccine development process for my public health project.
Hi, Maya. Of course. Tell me what you already know and where you need clarification.
I have a rough outline. Preclinical work in animal models, then clinical trials in phases 1 to 3, then licensure and post-marketing surveillance, but I am unclear about the focus of each clinical phase and the safety testing protocols.
That is a good start. Preclinical studies assess toxicity and immunogenicity in animals and support an investigational new drug application. Then regulators approve an IND and human trials begin.
So phase 1 is mainly safety and dose escalation, correct?
Yes. Phase 1 typically enrolls a small number of healthy volunteers. Researchers evaluate safety, identify immediate adverse events, and perform dose escalation to find tolerable dosing.
How small is small in phase 1 usually?
Often a few dozen participants. The exact number depends on the vaccine platform and whether prior human data exist. Early trials are cautious about severe reactions.
Then phase 2 comes next. I read that it looks at immunogenicity. Can you explain that term in this context?
Immunogenicity refers to the immune response induced by the vaccine. Phase 2 examines different dose levels and measures immune markers like neutralizing antibodies and T-cell responses.
Phase 2 is mainly about dose ranging and immunogenicity rather than proving full efficacy.
Exactly. The goal is to find the best dose and regimen that produces a robust immune response with acceptable safety before proceeding to large-scale trials.
What about phase 3 then?
Phase 3 are large randomized controlled trials. These are designed to demonstrate vaccine efficacy and to detect less common adverse events that smaller trials might miss.
How large are phase 3 trials typically?
They can enroll thousands to tens of thousands of participants. The sample size is powered to detect a predefined reduction in disease incidence between vaccine and control groups.
You mentioned randomized controlled trials. Is blinding always used?
Whenever feasible. Double-blind designs where neither participants nor investigators know the assignment reduce bias. Placebo controls are common, though active comparators may be used in some settings.
How do safety testing protocols work during trials?
There are several layers. Investigators collect solicited and unsolicited adverse events. Severity is graded. Serious adverse events trigger immediate reporting. An independent data safety monitoring board regularly reviews unblinded safety data.
What does the Data Safety Monitoring Board do exactly?
They provide independent oversight. The board can recommend pausing or stopping a trial if safety concerns arise, or if efficacy endpoints are met early.
Are there specific laboratory assays used to measure immune response?
Yes. Neutralization assays measure the ability of antibodies to block infection. ELISA quantifies binding antibodies. Cellular assays measure T-cell responses. Correlates of protection are statistical associations between immune markers and clinical protection.
You use the phrase correlates of protection. Are those always known before trials?
Not always. For some diseases like measles, correlates are well defined. For novel pathogens, researchers may identify correlates during or after phase 3 by analyzing immune responses in participants.
Can you give an example of an accelerated pathway?
During public health emergencies, regulators may allow rolling submissions and emergency authorizations. An example is the accelerated pathway used for Ebola and for recent responses, where prior platform data, plus strong phase 1 and 2 safety, allowed faster phase 3 start.
What about manufacturing and quality control aspects?
Manufacturing must follow good manufacturing practices. Consistent production and lot release testing are essential. Stability studies ensure the vaccine maintains potency during storage and distribution.
After licensure, what happens with safety monitoring?
Post-marketing surveillance, often called Phase 4, monitors rare adverse events and long-term safety. Passive systems collect reports from clinicians, and active surveillance studies can quantify risk.
How do trial designers choose endpoints in Phase 3?
Endpoints should be clinically meaningful. For an influenza vaccine, laboratory-confirmed symptomatic disease is typical. For other vaccines, prevention of severe disease or hospitalization may be primary endpoints.
Are there ethical considerations during trials?
Yes. Informed consent is critical. Trials must balance potential risks and benefits. Safety monitoring, transparency with participants, and equitable recruitment are ethical priorities.
What are common causes for trial pauses?
Unexpected serious adverse reactions, manufacturing issues, or clear futility in meeting endpoints can lead to pauses. The DSMB and regulators investigate and advise on next steps.
For my project, I need a real-world example. Could I use the Ebola vaccine development and the way Phase 3 was expedited?
Yes, the Ebola vaccine is a good case study. It used strong preclinical efficacy data, rapid Phase 1 and 2 safety and immunogenicity results, and then large field efficacy trials that were adapted to the outbreak context.
Great. For the section on safety testing protocols, I will highlight dose escalation, adverse event grading, DSMB oversight, and post-marketing surveillance. Anything else to include?
Also mention lot-to-lot consistency and stability testing, and the importance of validated assays for immunogenicity. And explain how correlates of protection can reduce the need for very large efficacy trials in the future.
Thank you. That gives me a clear structure. I will draft the paper and bring it to you next week for feedback.
Sounds good, Maya. I look forward to your draft. Remember to define technical terms for readers who are not specialists.
I will. Thanks again for your time.
Listen again to part of the conversation. Then answer the question.
Phase 2 is mainly about dose ranging and immunogenicity, rather than proving full efficacy.
This concludes the conversation portion. For more TOEFL practice tests and study materials, visit Spinta.org.